sector, the industry, the government, the nonprofit, whatever else needs to come in together to take on complex problems that don't have a single solution and require you to be agile and take risk. And we are all about that. So our bold vision, of course, is that we are recognized as the preeminent leaders in convergence research and being able to solve wicked problems. I think there are 5 words. We try to drill this into everybody that are define us. Integrative, that's in our name itself. Convergence, that I just talked to you about. Economic development at scale, and then making societal impact. And we want solutions deployed. And as you will hear today, this is all about how do you take things to deployment. So I want to introduce to you now, Dr. Théoden. I've never heard him being called that, Netoff. I've known Tate for, we were talking about it, more than a quarter century. When we were just starting, and in the field was just starting in the field of computation and neuroscience. And I think we've been really lucky that we've been in that field from the beginning of it.
Sure. So some of the things I want to point out is, University of Minnesota is a great graduate program. I don't know if there's any undergrads here, but we want to tell you. Renu and I have been running the neurotech workshop. This is a neurotech course, is a course that's online that we developed with the NIH grant. It's 21 lectures online, and it talks about how to translate technology from bench to bedside, and it goes through all the different steps. It's a self-guided course, but we have sprints a few times a year where you can, as a cohort, go through the course. And then we have twice a year a workshop where we bring about 30 people in for a 2-day intensive program where you develop and do a pitch. And so we're going to host that here, right?
Yep, in May.
But
it's an international group of people apply, and it is pretty competitive to get into it.
It is.
So especially the in-person one, it's very quite competitive. That it's paid for, right? Your travel's paid for, everything's paid for, but with which you come. So we will host that. We've hosted it twice in Minnesota, once in Denver and Colorado, and then May 2026, we will host it right here in this building.
Yeah, and we're very excited. So the one problem we find with people who do it locally is they don't appreciate what an opportunity it is. So they're like, yeah, I'll just show up to part of it. You're like, no, we fly people from all over the country to attend this, and it is a big deal. Like, you should be fully participating in it.
So if we can't do it, then we will have to. Got it? Okay, great. Thank you.
Okay.
All right. Sorry about that.
Okay, so if you think about the total number of possible settings, if you have frequency, amplitude, pulse width, and electrode, so we can turn any one of these on as anode, cathode, or off, how many patterns do you think there are? So do you think there's at least 4 patterns of stimulation? So raise your hand if you think there are more than 4 patterns. Okay, more than 4. How about 4,000? Keep your hands up if you think there's more than 4,000. Okay, some people are, okay, good. How about 4 million? Yeah, okay, good. 4 billion?
Sure, why not?
4 trillion?
I'm all in that one.
What?
I'm all in at this point. Keep going.
You're all in. It is 4 quadrillion possible settings. You have one that was left in. That's how many settings. You have about 43 million spatial patterns and then about the same number of temporal patterns. And you put them together and you have 4 quadrillion. And we hand this over to the doctors and you're like, oh, you can find the optimal stimulation setting for this and personalize it, right? Like, that's ridiculous, right? So if you went and tested 1 setting per second, it would only take 128 million years to get through all the settings. Now, we're not going to do an exhaustive search, right? But it is a real problem, and I think a lot of you guys are aware of this. So how many settings does an average physician test? It's like 2, right? So like 2 out of 4 quadrillion is, a small subset, right? And so like, what do we do to help these doctors test these things, right? So we have a large parameter space. There's a lot of potential for personalization, but how do you personalize this? So that's what I'm going to tell you about what we've done. I'm going to go and show you some examples that we've been doing in spinal cord injury, epilepsy, pain, depression, I've been thinking about it a lot in Parkinson's for a long time, but that I want to get through all these stories, so I'm going to rush really fast. So the spinal cord injury project started with David Darrow coming to me about 12 years ago and said, hey, there's this really interesting study where people have been implanting spinal cord stimulators. So this is the implantable pulse generator. It's threaded up into the spinal column. The electrodes are actually epidural, and it's been causing some, significant benefits for patients that are paraplegic. And so this is our first patient that we did. So you can see the spinal column here, and this is the injury. And you might not have to be a clinician, but this is not good, right, to see that, right? This person had a very traumatic, but there is still about 10% of the spinal column left there, only 10%, right? This person is motor complete. They have no sensation, no movement. They've been paralyzed for about 6 years. Here we threaded in the electrode paddle. This is the level of injury. So we're actually putting these electrodes below the level of injury, right? And here's a picture from us doing our tilt table tests. So you have the patient here, and we can measure their blood pressure. Usually we have something across the head, and then we tilt them up and their blood pressure crashes. This is Dr. Darrow here. This is Dr. Pena. And this is me. And this is Mel Brooks. And so here we have the blood pressure drops. We tilt them back, the blood pressure comes back up. We tilt them again and up, and their blood pressure drops. Turn on stimulation, and their blood pressure recovers. Right? So this is really quite neat. This is a really big problem for a lot of people with spinal cord injury. They can't get out of bed in the morning because their blood pressure is so low, because they lack the reflexes to squeeze the blood vessels to pump the blood back up to their head. So they can take a long time to get out of. But the interesting thing people saw was that it started providing and restoring volitional control of their legs. So here is a patient. She's been paralyzed for about, I think, 5 or 6 years. I'm going to show you the stimulation pattern we put on the electrodes and at different stimulation frequencies. And now it just occurred to me, I don't have control over this, but somebody has to press the go button on this. Who is controlling this cursor? There you go. So I'm stealing this from you. Okay, so there you can see. It's continuous stimulation going on. It's not a smart stimulation, and we asked her to move her leg. And you see, now we switched it to 50 hertz. It was much easier for her to move her leg. And then we go to 100 hertz because more is better, right? This is America, more is better. And 150% hertz, almost no control. You can see she's doing things with her other foot. I used to think that was bad, but when you are preparing to move your leg, what's the first thing you do? You stabilize the other leg, right, to do this. So you're seeing all of those behaviors. This is all completely volitional control. It's just continuous stimulation. So the question is, how do we find what the best setting for this patient is? So David, when he first came to me, said, I'm going to set up this study, and I'm going to give you the ability to program a patient once a month. And the device that we're programming has 15 settings on it. And you get to choose every month new settings for the patient. And we will repeat this for 1 year, and you get to optimize. So I don't know anything about programming spinal cord patients, and apparently nobody does. So this is what we came up with. So the first was we developed a Bayesian optimization approach. So let me just describe what Bayesian optimization is. So let's say on the x-axis is a parameter we want to optimize. So let's just say this is frequency for this case, right? And then on the y-axis is how good the actual control is of their leg, or something like that. So we have some measure of how well they can move the leg. We first start by testing 2 different settings. So one frequency, another frequency. We measure how good it is. And then what we do is we fit a line to those points. And one of the questions is, what's the kind of function we fit? And the thing about Bayesian optimization, use what's called a Gaussian process regression, and use what's called a functional. And so a functional doesn't actually fit a function, but it sort of fits a range that tells you any curve that fits within that range is actually a good enough description of it doesn't really matter, right? So we fit this Gaussian process regression. It fits tightly around the settings that we have, and it also estimates the standard deviation. And then you calculate what's called an acquisition function. So the acquisition function tells you how valuable sampling a setting in the parameter space is. So the acquisition function, the one that I like to use is called the upper confidence bound. So you take the mean in the black line and the standard deviation, which is shown, the range of it is shown by the blue, and you just add the standard deviation to the mean, and you find, and that's what this plot is down here, and you find the highest point on that, and you say that's the point I'm going to sample. So it's close to the settings that we already know, but far enough away from those settings that we're not exactly sure what the response is going to be, right? And so it tells us to sample that one. So then you go in and you actually test that setting, right? Oh, this one wasn't quite as good. We now fit the functional again, and then we go and calculate the acquisition function, and you can see it's going to tell us to sample there. On the next round, it's probably going to tell us to sample there. Eventually, it's going to tell us to sample here. But what it does is it very intelligently fills out the parameter space based upon every single data point that we have. It is computationally very intensive, but compared to doing an experiment on a patient, like it's not, it is very cost effective. You want to maximize all the information you have about these patients. So the first question, so we know what the parameters are that we want to optimize. So we have, frequency and pulse width. I'll explain why just those two. But we don't really know what we want to measure on that axis. So, what do we measure? Do we measure the motor skills? Do we measure things like blood pressure? But then we start realizing, there's all sorts of side effects that can happen. And I started going down the spiral of like, oh, all these things, how do we measure them? How do we measure all the things that they could benefit from? It just started becoming a big problem. But, so we came up with this, what I think is an ingenious, very simple solution, which is called a probit. So you guys have probably gone in, and a probit, what you do is you give two examples. And this is what psychologists have found is really useful, is you ask them only to compare two things. Which one do you like better, A or B? A or B? And that allows you to do some really difficult tasks like optimizing your glasses, right? That's a really difficult thing to do, but that system works really well. And so I'm going to show you how we do this in this case. So we actually program the patient up with a bunch of settings. And then and here I'm just showing you the frequencies that we did. And then we have them use a setting for a day. And they can use their cell phone to switch between settings. It actually communicates through Bluetooth to their implantable pulse generator. And they can switch between 15 different pre-programmed settings. So we tell them on day one, use one setting. On day two, use the next setting. And then go to a form and tell us, which setting do you like better, setting 1 or setting 2? So here they like 30 hertz over 50 hertz. So that was setting 2 was better than setting 1. Now we give them a new setting, is 300 hertz. We ask them, okay, compare 30 hertz to 300 hertz. They like 30 hertz over that. Now we give them a new setting, is 200 hertz. They like 200 over 300. So you get all these pairwise comparisons. And then you can actually build a model of the value as a function of this parameter space by making a model that best predicts these outcomes from the actual patient. And so it assigns an actual value on the y-axis to the stimulation frequency on the x-axis. And then you take that and you fit that curve with your Gaussian process regression, and then you start doing Bayesian optimization. So here, now we don't need a particular metric from the patient. We are using the patient to make those decisions. That's actually kind of difficult in some ways because the patient has to sometimes has to figure out what their values are, right? So let me show you, we've developed a really nice system for doing this. We have an app that fits on their phone that allows the patients to evaluate it. And we've developed this whole dashboard that is cloud-based so the clinicians can go in and actually select the patients, program all the patients, it sends up all the forms, it collects all the data, and then we can then make these what are called response surfaces from it. So the response surface, we have the x-axis is frequency, the y-axis is the pulse width, and the color is how much they like the setting. And so the pluses are the settings that we've already tested. We start off with a grid of 9 settings. And then once we've tested those initial 9 settings, then the algorithm analyzes it and suggests new settings. These are what we call exploitatory points, so points that are near, so these 3 settings are repeated for the previous settings that they were the favorite settings of the patient. We picked some settings that are close by, those are called exploitatory, and then we have some parameters out here that are called exploratory to sort of test the parameter space that we haven't seen yet. So once we've done this for a few months, here are what some of these surfaces look like. So here's examples from 6 different patients. These are some of our earlier patients, but we are now being really good at actually filling out the parameter space. We probably test more settings in a single patient than probably all the settings tested in all the other patients, right? And that have had spinal cord stimulators, maybe for pain and for spinal cord injury. And so one thing you should note is that if you look at these, they are different from patient to patient. Each patient has a different injury profile. They have, depending on where their electrodes are, they get different recovery. One question is, why do we only look at frequency and pulse width and not amplitude? Amplitude is dependent on the position of the spinal cord relative to the electrodes. And when they lay down, the electrodes, the spinal cord settles down, they have to turn down the stimulation, and when they stand up, it sort of separates out. And so they constantly have to change, depending on position, what the amplitude is. So we're leaving that as a parameter that they control. And so we're only optimizing over frequency and pulse width. So let me show you, this is one of our patients the day he went home. So this is, he'd been paralyzed for 2 years. We turned on his stimulator and his wife said, "Oh, honey, kick." And she joked that he kicked and she just almost broke down crying, right? And so they went home and reproduced that. But yeah, this is just continuous stimulation. This is that same patient you saw earlier. She was able by the end to do 30-pound leg presses, right, with one leg. This patient over here, she could actually time her kicks well enough to actually get some kind of walking action. Patients often want walking, but this does not restore sensation going up. But let me tell you a little bit about what we think is happening. So when you have spinal cord injury, normally there's a lot of inhibition coming down from your brain. So like if you try to ever do a patellar tap, right, and you do it on yourself, like I can never get mine to work, right? And that's because your brain actually knows exactly when that tap is going to happen, and so it actually prevents it from happening. And so you lose a lot of this inhibition, and your spinal cord becomes really hypersensitive, and it's sort of out of its range of sort of balance with excitation inhibition. What we think is happening when we stimulate, it actually causes antidromic activation down into the spinal cord, activating local inhibitory circuits, and it brings the spinal cord back into this balance of excitation and inhibition, bringing it back into its sort of working regime. And now that spinal cord can hear and respond to those signals that are still remaining from the brain. So, this is sort of the ideal thing. We're not trying to wire something from the brain into the spinal cord. The spinal cord is actually the best wiring, even if we only have 10% of it. It's still much better than we could probably put wires in and connect. And so we're really making use of those signals that are there. But, patients are interested in leg movement initially. But we find we get all sorts of other benefits from them. So, we started seeing you start doing quality of life measures. And so we didn't try to optimize for quality of life. We asked them what they like, right? And that can be different from patient to patient. So often they're looking at their legs movement, but then they start realizing there's all these other benefits. So quality of life and psychological health, we see improvements on that. But this is one of our patients that within about 6 months, she had been paralyzed for 21 years. And we put electrodes in. She had been catheterizing for 21 years. Within 6 months, she realized she had restored full bladder control, right? And she realized that she could pee without, even with the stimulation off, she could pee and control her. And she threw out all of her catheters, right? We're like, that's insane. But that became the most important thing to her is restoration of bladder control. We had no idea that was possible when we started this study, right? And so one of our patients, she came in and her husband sort of said to the doctor, was like, you know, keep setting 7. We really like setting 7. We're like, well, tell us a little bit about setting 7. Well, setting 7 restored her orgasm after 7 years of being anorgasmic, right? And so he's like, maybe you can turn it down a little bit because I'm kind of exhausted. But, you know, that became like one of the most important things to her, right? And bowel control, we start seeing patients that have bowel control. You know, these patients sometimes will take an hour and a half to 2 hours every other day doing digital evacuation. Digital meaning taking a finger and evacuating their bowels, right? That, and we see in most of our patients, reduces down to a half an hour, right? And because they get better core contraction, they get better control. An hour of every other day of not digging out poop is a huge improvement in quality of life, right? That is not something that people usually appreciate who have never participated in the spinal cord injury study. This guy came to us and said, hey, I think he'd been paralyzed for 17 years, and he came us 6 months into the study and said, I think I can move my legs a little bit with the stimulation off. And we're like, oh, that's cool. Turn your stimulation off the night before you come in. We'll put an EMG electrodes on you, and we will see if we can measure any muscle activity. So watch this. Watch his feet. See if you can see this movement. This is stimulation off 6 months into the study. I mean, that's pretty exciting, right? But then he did this, right? Like, you can see that, right? Like, that's amazing, right? So this is showing there's plasticity in the spinal cord. We don't know if it's like the patient is relearning the pathways. But, you know, being able to see some movement gives you feedback to really refine those, or whether the spinal cord is restructuring. We don't know. We can't do biopsies on these patients, right? We are stuck. We have small numbers of patients, right? We're about to implant our 27th patient, but we don't have any of them that we have biopsies on. So many unknown remaining questions. I want to sort of whip through this because I think these are the kinds of things you guys think about a lot. But, you know, when we do the electrode configuration, so I mentioned there's 43 million possible spatial settings. And what we do is we sort of pick some stimulation. We sort of start with anodes at the top and cathodes at the bottom, and we stimulate, and then we look to see kind of what functions that they're restoring. Do they get motional extensions or contractions or flexions? We look to see the balance between the left and right, and it sort of guides us through, and then we sort of lock in the spatial electrodes. But one of the things we're trying to do is automate this, because every doctor does a little bit differently, and we want to make a really standard approach. So we came up with a set of 42 different stimulation patterns to deliver, and then we go and we measure the evoked responses. So we put electrodes on the legs, and we measure the evoked responses with each one of those stimulation settings. Now, one of the questions is, can we develop a map of the spinal cord using that outcome that we can use to then prospectively design stimulation patterns to elicit specific muscle patterns, muscle activation patterns? So here we have our spinal cord with our electrodes in it. We have the muscle responses, and we're trying to make a map. And so what we did is, originally I was thinking, okay, we'll pick our electrode configuration, and we can select the potential map off of that. So we can calculate that fairly easily. But that doesn't actually solve it, because there's actually, depending on the orientation of the nerves, it depends on where it is relative to these potential maps. So we actually calculate an activation map. So we have an activation map, which is its second derivative of the potential in the x direction and the y direction. And we use that as our activation, where we think each electrode pattern is activating. And then we have our 16 muscles by 42 different stimulation patterns. And then we try to make this, we have our, given those stimulation patterns, these are the actual activation maps for all 42 of them. And then we have this map, which we're taking from our activation space back to our muscles. And so we develop a linear algebra model like this. We know that there's some We don't know what this map is. We know there's some nonlinearity into it, and so, but we can solve for this muscle activation map. And so here, when we do that, we can actually see for each muscle, we can see where the electrode, where the electrical potential, you would want to set it to activate that muscle. And it's complicated because these are going across multiple dorsal roots and things like that. And so you have different places in the spinal cord can activate the same muscle, right? So you get these complicated maps. But we can see that we can actually build this model on subset of these settings and then try to predict another setting. So we can see with different algorithms, but here are the different 16 muscles. This is how much activation we see of those muscles. And then I think there's a solid black line here, which is the actual activation when we do a pattern. And then we try to actually solve what it would look like given this model. So we'd say, okay, these were the electrodes that were being activated. Depending on the model, we can get pretty accurate representation of what we think that will look like. And then we actually did sort of a prospective study where we went back and we took a pattern that we had delivered, we measured the muscle activation on it, and then we said, okay, if we want this muscle activation, what is the optimal stimulation setting? The algorithm we use is sort of sign indifferent at this point. But it's slightly different pattern, but it's relatively close to this between our predicted optimal, and this might actually be more efficient than the settings that we use to elicit this muscle activation pattern. So this is something I'd love to talk to you guys a little bit more about and get your thoughts, because I think we have a lot in common. I'm going to switch stories now. I want to tell you a brief story about a chronic pain patient. This patient had chronic pain. She had, next slide, there we go. So she had a thalamic stroke, and she had chronic pain that felt like her face was cramping, and she had a level 8 out of 10 pain for 2 years continuously, right? And Dr. Darrow said, hey, we know there's different parts of the brain that people have tried stimulating for alleviating pain. One area, it turns out, is the motor facial area, not the sensory facial area, which was surprising. And the other one is the dorsolateral prefrontal cortex, which is involved in a lot of the perception of your emotional impact of the pain. And he thought, well, let's combine the two, right? We'll drill one hole, we will thread a set of electrodes over both those. And so we set down electrodes on her, and then we started into our Bayesian optimization, because I've got one hammer, that's my hammer. And we did a Bayesian optimization over her motor area first, and came up with electrode settings that she really liked, and some that she did not like. And then we did it again over dorsolateral prefrontal cortex, and then we tried to find a setting that worked for both of them combined. And when we turned that on after her 4th day, she said her pain was 0 for the first time in 2 years. She was completely transformed. It was really, really neat. We've done 7 of these patients. Pain is really, really difficult. It's worked well in some of them. It doesn't work great in others. We think there's a huge psychological component to pain. But these are, we're seeing a good impact in patients for which there is nothing out there for them, right? They are at their roadside. So my background is in epilepsy, and we decided, hey, let's actually try to do some of this Bayesian optimization in epilepsy. So one of the nice things in epilepsy is they have the Medtronic device is implanted, is FDA approved to implant into the anterior nucleus of the thalamus. And they have a setting that they use clinically, which is 145 hertz, 90 microseconds, 1 minute on, 5 minutes off, and you get a fairly good response rate. But is this the best setting for every patient? We don't know, right? And so let's go and try playing around with those parameters a little bit. And so one of the nice things that Medtronic developed was that this device not only delivers electrical stimulation, but it can record the activity as well. So Medtronic's Percept device we're using. And so here's our first, I think, 9 patients. No, we have 12 patients in here. No, 14, I see. so you can see our electrode positioning within. We sort of did some stats to look to see if we have differences in responders. This is the response rate, so baseline seizure rate. Baseline seizure rate, let me tell you, is a huge mess in itself. Doctors do not do a good job of really, establishing what the seizure rates are before they do these implants. So, take this as worth a grain of salt, but we look to see their seizure rates over time. We see some patients where their seizure rates go down a lot. These are our responders. These are patients, some response, and a couple patients that are not great. But let me show you how am I doing on time?
9:37.
9:37. So this is the kind of recordings we get in the clinic. So we actually go through and stimulate different settings. You can see the device is really cool because it knows when it's about to deliver the stimulation setting, so it actually blanks out the amplifier and then lets the amplifiers recover before it samples the next setting. So you get, even though you see a little bit of noise here from the stimulation, if you look at it in the spectrum, you actually see all that is very, very limited in the frequency domain of the stimulation. And you actually get really, really good imaging of, signal from the neural activity while you're stimulating. So we can look to see how the brain is responding to these different stimulation settings. And we start out with this initial 9 settings at different frequencies and pulse widths and measure how much activity we see with the hypothesis that settings that suppress activity might be better for seizure outcomes. The study we're doing, the first question is activity stimulation settings that we see suppress activity in the clinic seem to suppress activity at home. We're sort of leaving the seizure question aside initially. But we can go test those initial 9 settings in the clinic. We can download that data, and then we've developed a little MATLAB app that we can load in the data, and then it will create those surfaces and then suggest 5 more settings to test. So we will do 9 settings, analyze the data in about 5 minutes, do 5 more settings, and then we can program the patient, select the best setting, send the patient home. The device not only can record at 250 hertz sampling rate in the clinic, but at home, it actually measures the activity, and you have to specify a certain frequency band, but it will measure 10-minute averages of that power in that band continuously for 2 months, and then you can download the data. So, it is only 144 samples a day, but you're getting 144 samples a day of their brain activity for months on end. And we go and we download the data every 2 months, and so we get we have almost continuous recordings from some of these patients. I'll show you in a second. But so here are examples of when we look across all of our clinical visits for 2 different patients, what that response surface looks like. We see what setting we set them home with, sort of based upon. It's kind of noisy when you just do 1 clinical visit. So we sort of selected. It's not the worst. It's not quite the best on those patients, but and then when we stimulate, you can see some of these. This is what the baseline. We see this very strong. This is a power spectrum. Sorry, and we see a very strong gamma rhythm in these patients. And when we turn on the stimulation, that gamma rhythm really drops. And so you can see in our responders, this gamma rhythm is really strong, and it drops a lot. And then you can see in the non-responders, they did not have very much gamma rhythm, and it does not change very much, except for this patient here. This patient we eventually got to respond because we found settings for her that worked. Here are some examples of, so we think that we found a biomarker of patients who are going to respond. If they have this strong gamma rhythm, we think they're going to be responders. And we're now going to go and test that hypothesis prospectively by doing stereo EEG recordings from the patients, looking to see if they have these gamma rhythms, and then looking to see if they have a good outcome. Because if we can identify patients that don't respond to the therapy before they get the implant and sort of remove them, that can really improve the response rates, right? By removing those. Ideally, we'd find a solution for them, but at least knowing that they're not going to respond is useful. Here are some of the recordings we have. These are that 10-minute averages. So you can see here are a couple months straight of those. Here we switch settings. So you can see as we switch settings, you see a significant difference in the amount of brain activity from one setting to another. I'm just going to run through these really quickly. But we have over 20, I think we're up to 28 years of recordings of data from our patients. Some of them we have almost continuous recordings for like 2 to 3 years. And you can see how things can change over time. You can see these are labeled by day and night cycles. So you actually see, if you calculate the power as a function of the hour of the day, you actually see strong diurnal cycles in these patients. Sometimes you see significant increases in that activity before the onset of seizures. And so here are those patients that the strong gamma rhythm, those gamma rhythms can change over time. And then we've been looking to see, here are responders, I talked about those before, and we found that those responders have these gamma rhythms, at least in one hemisphere. This one's a little small, and then here's that one patient that we thought should be a responder. We did get them to respond eventually, but the other ones that were not responders did not have that gamma. But if you look at the cycles of this power over time, we actually find that it varies a bit. So these are from visit to visit, what their power spectrum looks like. And sometimes their gamma is strong and sometimes it's slower. And we actually find a fairly strong correlation between their seizure frequency and the amount of gamma activity we see in their brain. So if we see a suppression of that gamma, and so that gives us further evidence that we should look at sort of trying to drive that gamma down. And then here is looking at that power over time, so we can set, initially we were looking at alpha power, and so this data is only showing the alpha power, but this is from the 10-minute averages of that alpha power over time in a bunch of different patients. And this is a wavelet transform, and you see here is the diurnal cycle. So you see strong diurnal cycles in most patients, and then we see other peaks in that. But what we see is that seizures in a lot of these patients, so if you look at these histograms and circular histograms as a time of day, we see all their seizures happen in a particular time of day, right? And so you can look at either the phase of the neural cycle or the actual time of day, you see strong cycles. And one of their questions was, does seeing the variation in the neural signal, does that help inform us and predict better than just looking at time of day? And in fact, it seems to give us marginal improvement in our seizure prediction accuracy. So we also can combine multiple cycles. Sometimes you see peaks at like weeks or months long, and we combine both daily cycles and weekly cycles to see that there are times when both those coincide and you get days that are much more likely to have seizures at times of the day than at other days. And you can combine those, and we found that we get, not great seizure prediction accuracy, but better than, by chance, using just these neural cycles and looking to see when seizure. And we're looking at very small numbers of seizures in these, but you do see strong phasic locking of these seizures to these cycles, and you can combine multiple cycles for seizure prediction. And I think I have just a few more minutes. Here we go. So I want to tell you the patient that we had for depression. This is a really interesting study. This is our first patient. And this is a collaboration with Ziad Nahas, Robert Hermosillo, Damien Fair, and then David Darrow was the neurosurgeon and put him in. So this patient had really severe depression since he was like 13. Several attempts at committing suicide. He's undergone 26 failed therapies, including many different drugs, but ECT and TMS. And so what we did is we started by mapping out his brain. So Damien Fair has this what's called precision functional mapping. Takes the patient, does like a 40-minute functional MRI on their brains, and then they look at every voxel in the brain, look at correlations with all the other voxels, and looks for networks. And the idea is you can see certain parts of the brain all participate in different networks. So we have like default mode network, the action mode network, salience network, and the frontal parietal network. And we saw this patient had a very strong salience network. So using these maps, we place electrodes down over the brain targeting some of these networks with the hypothesis that modulating some of these networks might actually help alleviate some of this depression. And so these are the same networks that other people like Helen Mayberg has been doing, but at different positions within the network. So they're going down into, was it subgenual? ACC, yeah, thank you. So it's an ACC, all of these pathways converge there, but we can also access them from the surface of the cortex. So we implanted this patient, and in the ICU, this is my cameo here of my laptop, but here hopefully we can hear him talking about it as we turn on this combination of electrodes.
It's you can just sense the waves, the wash.
Because that smile.
It's the first time we'd seen him smile in 4 days.
It's emotional, huh? What is making you cry with him?
It feels good. I don't know what to do with it.
Well, hopefully it's going to become more habitual.
Yeah.
I think it's true.
Let's sit it for a second.
Can I,
You saw just profound changes in him after this stimulation.
Sadness and pleasure and all
When I talked to him afterwards about it, he said it was like he had been a lost child in the grocery store for all of his life. And he just sort of come to the conclusion that he was going to be a grocery store boy for the rest of his life. And then his parents come around the corner and you see his parents. He was like, that's the feeling he had when that stimulation was on, right? And he said it was just, and he said, like, you know, I was talking to him afterwards, like, you've gone through a heroic amount of like brain surgery and testing for 4 days. And he was like, you know, even if I never feel that feeling ever again, having felt it once in my life made all of that surgery worth it. So, you know, really exciting result initially. And then what we did is, you know, they turned it over to me and like, okay, Tay, now you've got to find, you know, the stimulation setting out of 4 quadrillion. So, you know, we started our Bayesian optimization and testing at different frequencies and pulse widths. We had them test a setting for 3 days because it takes a little longer for these to, we don't know exactly the right amount of time to test a setting. We're still trying to figure that out. But this was we had 4 months to program and we were only able to test 5 settings in a month. And so we went through, this is at the end of our 3rd month and our programming for our 4th month. So we saw he really liked these settings. So we were doing our exploitation settings here. And then, you know, we had 1 exploratory setting. You know, we don't know what's going to happen here. We put that there. And he came back the next month. He was like, I love this setting. And we were like, wait, really? And we're like, okay, well, we'll send you home on this setting. So, you know, we actually monitored daily his depression scores. And so we could actually look at his depression scores. So as we were optimizing, we were testing, so the scores came down a little bit, but they're fluctuating around. We finally locked him in on that setting he really liked at the end of 4 months. And then you sort of watch his depression scores continue to drop as he starts to really improve because of the therapy. So it's not a setting we were expecting. There were a lot of things we didn't expect. We didn't think changing the pulse width would really have an effect at all. His favorite settings were here. We increased the pulse width by 50 microseconds and it became one he really hated. That seemed really surprising to us. But this is one of the beauties is like we don't bring in a lot of, you know, preconceived notions. I don't bring a lot of preconceived notions of what's going to happen, right? And so the data says that, well, there's some kind of cliff there. We're just going to avoid it. So we are now, we're in the middle of programming our 2nd depression patient. She's not responding quite as well as the first patient, but we also haven't locked in the settings yet. In about a month, we'll lock her in. And we'll hopefully get into this phase where we start seeing her drop and really get better. So take-home messages, we can use neuromodulation for a lot of disorders, right? So Parkinson's and OCD and, we're seeing it in depression, but there's lots of applications. The idea of having all this parameter space is great, right? And, I see people saying more electrodes are going to give us more opportunities, right? Larger parameter space, more opportunities. But at 4 quadrillion, you're asking, well, really, how much do we need, before we start? And so Bayesian optimization and particularly probits have been really useful for us. So in the epilepsy space, we're finding biomarkers. We actually have the neural signal. We're using the neural signal. But for the depression and for the spinal cord injury, probit has allowed us to discover things that we would not have before, right? Because we're asking the patient what they like. They get to use their own values to decide what is good and what is not good. And I think it's been really, really important in getting the patient engagement, empowering them, and discovering things that really benefit to them. And so I really like this approach. I think it's really simple and very powerful. I want to thank all of my lab members. So Zach Sanger now has moved to Medtronic, is now working at Medtronic. And let's see. And then, David Darrow and Bob McGovern and Andy Lampersky is the controls engineer who sort of taught me everything I know about control engineering. And then, of course, my funding that has supported this. And we've had a lot of really great funding for the Folks Research Foundation in supporting some of the early stage of the depression studies, and the Minnesota Office of Higher Education. It's a state-funded program that has supplied a lot of the funds for our spinal cord injury studies. So I really appreciate those. So. And then one last thing, the Minnesota Neuromodulation Symposium. It has been a spring conference, but we are now breaking it away from the Design and Medical Device Conference, and it will be in October of next year. So it'll be about a year from now, and really hope some of you guys come up and see it. So I think that's it. I already did these slides when we were killing time at the beginning. Okay, that's it. Thank you.
Fascinating. I'm sure you have questions.
Yeah.
Hi, amazing talk. I really enjoyed it. So I had a question regarding your Bayesian optimization that you do. It looks like from the last slide you showed, sometimes you get to the bias solution. How often you get to the bias solution when you're doing the cross patient?
Yeah.
And maybe the second question after that, have you tried Bayesian with hybrid optimization, like at the tail of it, do like quasi-Newton optimization to get to a better solution? Have you tried with those things? I was wondering that if it works or not.
Yeah. So there's a lot of control theory that really. So when you do it in a computational model, you have time to really look for convergence and things like that. And unfortunately, in the real patients, we don't really have much time, and things are changing often so much that I'm not saying that we are finding the global minimum. What it is doing is empowering the physicians to feel like they have a structure to do exploration, getting feedback, and testing new settings. I don't think in any of our patients have we gone far enough to feel like we've converged. And you're right, there are so many tools that we could use. Especially things like hierarchical modeling, where we start taking patient data across all the patients that we've done when we have more than 2. Maybe we're approaching that with spinal cord injury with 27. But there's a lot of tools I think we can expand using, especially things like SafeOpt, where you start off with a fairly small parameter regime. Especially in epilepsy, they were like, you can tweak it by 20%. And then as you test settings, you get out to the edge of that parameter space, you expand out because you already know that it's relatively safe, you can keep expanding out there. So I agree, there's a lot of tools. I do not try to tell people that we are converging to a global minimum. I think it only as an empowering of the physicians to do a parameter space exploration and gives them support and some kind of feedback. I'm not claiming anything more than that. Lots of opportunities.
Well, Ronald, amazing presentation. I have one short comment and one question. My short comment is this: I truly appreciate that you put the photo of your colleagues in each section.
Oh,
that
good.
is really nice.
Thank you. Thank you.
Thank you on behalf of them. Thank you. My question about the take-home message is that 40% of older adults in the United States are suffering from physical mobility due to brain neural issue, arthritis, balance and control, and muscle loss. Do you think any future of your program is, a state of art to help older adults in combination with physical exercise, physical therapy to improve their physical mobility? Is any study is done in the United States, or am I talking totally nonsense?
No, I think you're exactly right. And I see this as like my one hammer, right? Like, of course it could be used for that. I've seen some really nice Bayesian optimization applied to using like assistive robotics and looking at the energy consumption, whether it actually is improving the patient's energy consumption or not. So I think, if you have a therapy and there are parameters to that therapy other than just like drug dosing, there's a lot of opportunity. And I think you're right. Like those are the applications that could have huge, huge impact. I don't know whether we will get to the point of actually implanting electrodes to support mobility, but it is, feasible. And I think we're seeing a lot with post-stroke patients. They're putting in stimulators, and I think there's a lot of opportunity there to do optimization with electrical stimulation.
Thank you.
Yeah, that's exactly right.
Yeah, thanks a lot, Tay, and go Bears.
Thank you.
So I'm always trying to figure out how you back out from this type of work applications to basic psychology experiments.
Yeah.
And it reminds me in TMS, transcranial magnetic stimulation, lots of issues and just heuristics that people use for repetitive TMS, where you get the motor revoked potential and you go 70% of threshold and just apply that at 40 hertz to virtually a lesion or break lesion.
Yeah.
Right? And probably where Rich Ivory and Cal was doing this in the early 2000s.
Yeah.
So how would you apply your insights from this world, and maybe even just getting a better read on the motor revoked potential, to then tell TMS people, here's how you would stimulate the DLPFC to virtually lesion?
So one that I've thought about, I work next door to Alex Opitz, and so Alex is working a lot on like phasic stimulation relative to neural signals, and actually doing computational models of looking at the anatomy and the interaction with it. But I think looking at those evoked responses and using the enclosed loop to optimize the therapy, both the orientation of the coils as well as the frequency, pulse width, and those are all parameters that should be optimized. So I totally agree. It's another opportunity. Yeah.
This is no formal link with, for example, tragically Nolan Williams' Saint procedures.
Who tragically
last
passed
week.
away last week.
Any formal connection between what you're doing here in parameter optimization of below the spinal cord with Saint procedures or depression?
So one project we have, I'm digressing a little bit, is one of the questions always people have with the volitional control is whether it's actually cortically controlled or whether some way patients are faking it. So one of the things we talked with Alex Opitz is, can we do transcranial magnetic stimulation, get an actual evoked response in the leg, then add in the stimulation and see if we can actually modulate that to show that it's actually cortically controlled. So that's one thing. And then if you can do that, of course, you can start optimizing by looking at just the size of the response and trying to maximize that. To hopefully then your actually volitional control will be improved. But with TMS, we've gone down to the cortical stimulation. So the nice thing with cortical stimulation is we have much more control spatially. But Alex Opitz and Damien Fair have a big study going on looking at using TMS and the personalized functional maps to guide the stimulation. So I like the electrical stimulation just because we can get down to the cortex. It's much more invasive, right? But it gives us a level of precision. But these things exactly, carry over from our electrical into TMS.
Yeah, we have a question from the room, or from the virtual room, Matt, Judah. In the depression patient, it seems that some of the beneficial effects were seen not just at 20 hertz, but also at the harmonic frequencies of 20 hertz, e.g. 40, 60, etc.
Yeah.
What do you make of that?
Great question. I have no answers. Yeah, I mean, this fun thing about these things is that they raise more questions than they answer, right? Yeah, and I come from a background of phasic stimulation and looking at enhancing or decreasing certain oscillations in the brain. So this is one of the exciting things about finding this gamma rhythm in epilepsy is that now we can use some of my old theories of how to do periodic stimulation that modulates those oscillations. So I came up with the hypothesis of chaotic desynchronization. So there's frequencies that if you drive an oscillatory system at those certain frequencies, you get chaotic behavior, and then that causes the system to desynchronize. And so there are times you get these Arnold tugs where for certain frequencies enhance the oscillation and sometimes it destroys the oscillation. And so I agree, seeing that kind of harmonics at the same frequency gives me some feeling like that maybe we are interacting with an actual rhythm in the brain, and we can constructive or destructive or even chaotic desynchronization of that, and we should make use of it. But we need to be able to see those neural signals in order to actually design stimulations to do that. And so this, of course, is just the beginning, right? The Bayesian optimization is a fairly black box approach. With epilepsy, we're starting to see the signals, and we have a lot of background, and Jimmy and I were talking about all of these control theory applications once you start getting in the loop. And the guys with the TMS are starting to do that phasic stimulation, and I have lots of thoughts to go from there. So I'm not done with my career yet, hopefully.
Do you tend to see changes in how people respond to the stimulation over time? Or is
Oh my God. Okay, yeah, so this is the dirty secrets. It's really messy, right? So I have patients in the spinal cord injury population, they are convinced that setting one, I would go in while they were at home and reprogram them. So setting one was a different setting the second time they used it. I'm I swear to God, I'm not changing anything, right? And so they can be, then they can tell me also oh, I love setting number 4 from 3 months ago, but I've never had a setting like setting number 4 ever again. And then you say, well, I always give you your top 3 favorite settings. I repeat those every time, but I change the number on it. And that setting 4 from 3 months ago is now setting 6, right? And you've actually had that the entire time, right? And so it's what is changing, right? I don't know if it's the spinal cord is changing, if the patient's perception, patient's expectations. I don't really know, right? It is a difficult game of one of the nice things, we have these programs that we change the program number on it so they don't know, it's semi-blinded, so they don't know what setting they're using from month to month. We have had patients, when one of our first pain patients that we did, she said, oh, I love this setting, but about 3 months into it, or 3 days into it, I get this flare up, the pain is unbearable, turn off the stimulation, so we turn it off, then she comes back the next week, we reprogram her, and so over a couple months, she said, okay, I'm getting much, much better, I'm gaining much, much more long time that I can stand the stimulation. Just bear with me, I'll push it out more and more. The thing is we started her on a sham stimulation, and we had never really turned it up above 0.5 milliamps. And so we realized all of that was psychological expectations, right? So you have real problems of, we were hoping that would extinguish, and then she'd be okay, now I can handle that stimulation for, weeks on end, no problem, right? And now we can start actually optimizing the therapy because we've disambiguated her expectations and from the actual therapy. But we eventually had to sit her down and we're everything you've experienced so far is, nocebo effect, right? Is not actually from the therapy at all. So I don't know if that answered your question, but it is very complicated. And that's one of the reasons why, how much can we really optimize and apply these really fancy optimization techniques? Really, the problem is that there's so much variability in the patient at this point that, I don't have a lot of real promises that we're finding the optimal solution in the short time that we have.
Question. So it sounds like, obviously, you mentioned it right there, there's this tremendous variability within the patients.
Yeah.
Have you guys found anything that you're hey, this just seems fairly universal that's going to basically improve sort of the basic use of these for all these patients that don't get to go through this optimization procedure?
I mean, I can't say that I have, right?
So how are they finding these initial settings? You know, I mean, you sort of say, hey, this is what they do.
Yeah.
How did they come up with that number, those numbers?
So like with the epilepsy one, there was the standard clinical setting, and we convinced the epileptologist, we could tweak these numbers by 20%. Would you be comfortable with that? And they're like, okay, 20%, that's fine, right? So that's how we started with it. With the spinal cord injury, like, you know, we go from, you know, 260 microseconds up to 500 microseconds, right? And then we have ranges we thought kind of work for the autonomic settings. I didn't talk about, we actually have separate autonomic settings than the volitional settings. But yeah, it's a big question, and especially if you only get maybe 7 or 8 settings, right? Like, are you going to make them really cover the entire parameter space, or are you going to sort of like, we don't know how smooth it is, we don't know how long they really have to be, especially with depression, we really don't know how long. I've come up with algorithms where it's called BALD, where you actually, every minute we ask the patient to provide a score on how they're feeling with the pain, and then, or depression. And then the algorithm tries to make a model of how much response they get with stimulation and a time constant of the onset and the offset. And then what it does is it then selects which setting to deliver on or off according to which model it's less confident about what the outcome is going to be. So it actually optimally searches to provide this information for our data. So, but when you only get 20 minutes of recording, you don't know if that's gonna. So next Friday, we're gonna test this in our first depression patient. We've done it once in a pain patient. I'm not sure the results are convincing at all. But these are the basic questions we have. We're just at the start.
So how big is the market? How many of these things are getting made in the US?
Well, for the chronic pain patient is quite a big market. So there's about 400,000 people implanted with the chronic pain for spinal cord stimulation. That one we kind of feel like is the biggest one if we're going to do it. There's a lot of people with vagal nerve stimulation. So I've been talking to Livanova that does the vagal nerve. Like if we could start doing some Bayesian optimization for epilepsy or depression in their studies with those devices, I think that would be the biggest market. Parkinson's, I think, is a big enough market. So I've worked with the big Parkinson's team, but they have not yet adopted all these tools. I think they're quite interested, but we need to get it into our clinics. And then epilepsy, it's hard to tell whether epilepsy is a big enough market. Like neuro stimulation is not the market driving for these big companies. Like diabetes and the insulin pumps, right, is what they make all their money. Spinal cord stimulators, we're trying to convince them that there's a big enough market for spinal cord injury patients to be worth it. One of the big things is the entry into the market is relatively low because we're using devices that are already developed. So they don't need $1 billion to break into this new market, right? They just need to give us 30 million. So,
How about the substance?
this is the one with pain. We're hoping that the impact on substance abuse would be great. Because we think a lot of the substance abuse is driven by pain. That's one of the routes that we're hoping to justify what we're doing. Yeah. Oh my goodness! I didn't realize there were going to be so many questions.
I think okay, I'll start. Yeah, I have a short question. So I'm an econometrician, so I use like a lot of logistic modeling and like probit models and as well. So interesting choice of like optimization technique here. So just curious to know like how big was the factor of like computation speed, time and resources versus like the accuracy of the model, and was that an active choice?
Yeah. So that's a great question. As far as the probit, and when we're talking about the number of settings that we have here, it's computational time is not. Like it's not, I think when people are thinking big data, right, it doesn't apply well to big data, but this is not big data. When we do some of the animal models for epilepsy, we will go through and test like 1,000 seizures over like 4 or 5 days. Then we're starting to get up into data sets that are a little bit large enough that it's starting to take, on the order of the time between seizures that we see to do the analysis. But here, we're not seeing anything. And you're right, accuracy is more limited by the data than it is by the methods. Yeah, good question.
Mine is more of a curious question. How long after surgery have you found is the best time to start your stimulation optimization?
Yeah.
And how does that vary across the conditions that you're testing?
Great question. So part of it sort of depends on how much we think that you need to recover from the therapy. So with the spinal cord injury patients, we can bring them into the OR at 7 in the morning. By 11 a.m. out of anesthesia, turn on stimulation, they can move their legs, right? Within like 4 hours, right? Sometimes we get the best movement immediately post-op, and it's not as good as a month later. But we know that there's a lot of trauma when you're putting it in, so we kind of want everything to recover, so we don't really start the optimization until a month afterwards. And that's kind of practical choice with depression patients. We put the electrodes in, and then the next day we start our optimization for at least electrode selection, right? And we're doing psych tests, and these guys, like this first depression patient, he was still kind of throwing up, and we're like, okay, test this setting. He's like, oh, I don't feel so well, and you're like, the 4 days we had in the hospital cost us $350,000 in hospital bills, right? So, it's like, dude, you're gonna start now. So I don't know. It's a good question. I don't think it's ever too late to start. So.
I just want to note that we're a little bit over.
Yeah, we're over time. If people need to leave, I'm not offended. Yeah, thank you for staying to me.
I have a quick question. What was the most surprising side effect that you found for like the therapies that you would like mention?
Orgasm. Yeah. Yeah. I mean, orgasm was one. The bladder control, that was really surprising. So the most surprising has been and then the bowel control was really surprising. We've also seen significant improvements in sexual function. So we're now doing sexual function study. We have a whole laser Doppler system to measure blood flow in the genitalia while I have patients looking at erotic videos. We finally got our first male in and then had him watch erotic videos, and we saw these waves of blood flow go across the scrotum as he's watching erotic videos. It was like, what the hell is that, right? And so like there's, I mean, just if you want to know what the most surprising things are, yeah. Yeah, did you?
Well, it was going back to Aaliyah's question, but with our sensory project that we're doing
yes.
with the hand.
You can see why I'm excited.
Yes. We caution like stimulating before like maybe 3 days after, or like maybe I think one patient might have been 2 days after surgery. But then we've seen that sensation doesn't seem to stabilize, like how they're feeling it until like maybe 3 months to like, but it still can kind of change over time.
Yeah.
And our theory is like, the nerves basically calming down, inflamed and everything, and then scar tissue potentially building up. Have you seen anything where like, rather than just results, I'm thinking like the depression patient, but like rather than in like the spinal cord, anything like that where it just kind of stabilizes over time because of maybe things healed or?
I think you have a much better readout than we do, right? And so like, I think you're able to quantitatively see this better than we can. With the spinal cord, our readout is so, we can see motor movement, we can measure that pretty well, but we don't really see a whole lot of change. We're going to some fairly sophisticated methods to look for changes over time. We don't really know what's causing those changes. We are looking at impedance, changes of the electrodes. These are bigger macro electrodes, but I think I'm more informed by your studies than I can inform your studies. So,
are you doing any imaging to like? What methods are you planning to use to look at that? Like, is there specific imaging that seems to
Unfortunately, with the hardware we have in them, we cannot do imaging after implantation.
Oh, okay.
So like we do functional MRI in our depression patients and our epilepsy, or we do MRI in our epilepsy patients before, but with all these electrodes, you can do some imaging in other parts of the body, but you can't do imaging at where the electrodes are, which is where we're interested.
Have you tried anything with ultrasound?
So I have a whole ultrasound study, I could give that talk too, but that's older work, and I would be interested, we're having a discussion last week about whether a non-invasive neuromodulation lab should buy an ultrasound, focus ultrasound. Yeah, we did a whole study in animal models, which I'd love to talk about, but yeah, but no, we have not done any focus ultrasound studies, and that could be a possibility.
Okay.
Yeah.
Thanks.
Love your ideas on that.